I apologize. Okay, so these filaments are going to slide over each other in a sliding filament model. So how does this happen? We have a link between the action potential in a motor neuron and the skeletal muscle contraction. We call this excitation contraction coupling. So acetylcholine is released from our motor neuron. It binds to the receptor on the muscle fiber. That then depolarizes our muscle fiber. That depolarization travels down the T-tubule. It causes the release of calcium from the sarcoplasmic reticulum. The calcium releases the block on actin. Myosin head groups are able to grab it, bind it, pull it to the middle. I'm going to repeat all this with a picture. I'm just putting this into words. Exactly what I said, but now with a picture. Let's follow along. We have acetylcholine. Acetylcholine is released from our motor neuron. It binds to acetylcholine receptors on the membrane of our muscle fiber. It depolarizes our skeletal muscle. That action potential will travel down the T-tubules. When it gets near the sarcoplasmic reticulum, that will trigger the release of calcium. Calcium is going to bind to troponin, which is going to expose the binding site. Myosin head groups will grab onto the actin and pull it to the middle. And this will keep occurring as long as there is calcium. Eventually, the calcium will be transported back into our sarcoplasmic reticulum. Eventually, the acetylcholine will be degraded. This whole system will be turned off. Okay, this is our skeletal muscle. This is theoretically a review. Now, for our heart does not need a signal to contract. The conduction system of the heart is an autorhythmic system. So these cells, specific cells in our heart, have the ability to generate their own action potentials without input, without a signal. And these cells we have provide a pathway that spreads that excitation throughout the heart, that electrical signal, that depolarizing signal. Cardiac contraction occurs. We're going to be thinking about two different types of cells. So we've got cells that depolarize spontaneously, and we have our pacemaker cells. So our pacemaker cells have an unstable resting membrane potential. So if we remember that diagram that I drew 500 times for a neuron, right, where we have resting membrane potential for neuron at around minus 70 millivolts, it takes a signal to bring it to threshold at negative 55, and then it will fire an action potential. In our pacemaker cells, they don't stay at rest. They are continually approaching a threshold and then firing an action potential, approaching a threshold and then firing an action potential, okay? They do not have a signal. They generate their own instability. We also are going to talk about cardiac myocytes. These are connected to pacemakers by our gap junctions, and these are the ones that do the contraction. So we're going to have one set of cells that generate this action potential, and they're going to pass it to a second set of cells that will contract. The pacemaker cells are what control our heart rate. So if we think about our heart, again, that contraction is going to occur in a rhythm. And the rate at which the pacemaker cells fire their action potential, that is our heart rate. And the fastest cells in our heart are the ones that will set the rhythm for the whole heart because they will fire first. And these are pacemaker cells specifically located in an area of our heart called the SA node, or the sinoatrial node, which is in our right atrium. Again, pacemaker cells don't contract themselves, but they set the rhythm. So we're now going to talk about action potentials in pacemaker cells. This is a confusing section because we've talked about action potentials in neurons. We're going to talk about action potentials in pacemaker cells. And then we're going to talk about action potentials in our cardiac myocytes, the cells that contract. All three of these cells have their own flavor of action potential. They use different channels. They have different ions. They have different values in terms of membrane potential. They have different slopes, right? But they all follow the same general principle of depolarization to repolarization, okay? And so I really want to make sure that we are clear when we talk about these three different potentials, okay? We're not going to get overwhelmed. The numbering is weird. You're gonna look at this and be like, excuse me, professor, I think you made a typo. That should say like 123 instead of 403. Yes, theoretically, theoretically it should. Don't worry about the numbering. The numbering comes from the cardiac myocyte. So cardiac myocyte action potentials define the numbering, and everything else just follows it. And I'll show you what I mean when I say that when we talk about cardiac myocytes a little bit later. So we're going to start at baseline in our pacemaker potential. This is phase 4, and this is unstable. So you'll remember in a neuron, here I would have drawn like a straight horizontal line to be like, we're just at rest. You'll notice right away that that doesn't happen. We are constantly at a slope, a slightly small slope of depolarization in phase 4. And this is brought on by a few channels, by 3 channels in particular. We've got a channel called a funny channel. Now, I didn't name this. It's a weird name, but they call it a funny channel. And this is our non-selective cation channel. Okay, let's remember back. Cation channels, positive things, whichever ion has the strongest driving force wins the day. So at around -60, sodium wins the day. This is a depolarizing channel. So at rest, the funny channels are open. So sodium current, that's what this P thing is, the current for sodium, is on the rise. We have our potassium leak channels are closed. This is different from a neuron. Okay, we're comparing and contrasting. This is different from a neuron where at rest, potassium leak channels are open. The other channel open are a specific type of calcium channels called our T-type calcium channels. But in general, the pacemaker potential is set by our funny channels. Okay. Then when it reaches threshold, which is around -40 for our pacemaker cells, we're going to have a slow action potential. And this is driven here by calcium. So we're not talking about sodium anymore. We're talking about calcium. And this is through a channel, which is a voltage gated channel called our L-type calcium channels. They're going to open and our cell will depolarize, but it's less steep than in a neuron. I know it looks really steep here. Take my word for it. It's less steep than a neuron. Then at phase 3, the calcium channels close. They reach equilibrium potential for calcium. They close, and we open a specific channel called a rectifier potassium channel, and that will repolarize our cell. The time frame here is super, super important. We're talking 200 milliseconds. For a neuron, an action potential is around 2 milliseconds. So this is 100 times slower, okay? 100 times slower. I'm going to do the same thing in a different picture. Oh, no, we're going to break into groups. Okay. I'm going to break you guys into groups, and I want you to discuss this question very briefly. What would happen to our pacemaker potential if we opened more funny channels, if we opened more T-type calcium channels, or if we opened more potassium rectifier channels? Okay, I'm going to break you guys briefly into groups to discuss this question. Into 3 groups. Okay. You should get a little notice to go into a room, unmute yourself, and talk about it. Shimon, is everything okay? There we go. Okay. People are coming back, coming back. I know that was a very brief time. See if the other two rooms are coming back in about 30 seconds. Okay. Give them their last few seconds to chat. Last room. Okay. Last few seconds. So when we think about these questions, right, what would happen if we do something to a potential, we really want to think about where in the action potential those channels are involved. What are they involved in? Okay, so when we think about the answer to this question, we want to think about what phase or what role each of those components has. So let me bring my PowerPoint back. Give me 1 second. Let me open this up. Oh, I didn't mean to click play from start. Oh, that's fun. Give me 1 second, guys. Computers are fun. Play from current slide. Okay, so when we want to think about a question like this, we want to ask ourselves, what is the role of each of these channels? Okay, so let's go back a slide. Let's go back a slide. Our funny channels and our T-type calcium channels are both involved with setting our pacemaker potential. They're both involved with this slow rise to threshold. So if we open more funny channels and we open more T-type calcium channels, those are both depolarizing effects. What we're going to do is we're going to increase the slope of our pacemaker potential. So we'll take it from a shallow slope to a much steeper slope. We will, in effect, reach threshold faster. Okay, we will in effect reach threshold faster. And if we think about this in the long term, normally at baseline, say in 200 milliseconds, we would fire one action potential. So in the course of 400 milliseconds, we would fire two. That would be 2 heartbeats in effect in 400 milliseconds. If we reach threshold faster, we can fire that action potential faster. And so say instead of 200 milliseconds, it now takes us 100 milliseconds. So we would, in a period of time of 400 milliseconds, fire 4 heartbeats as opposed to 2. We've raised our heart rate, all by opening more funny channels or opening more T-type calcium channels. If we open more rectifier potassium channels, now we're affecting phase 3. And we can also, in this effect, increase the slope here. And it would have the same overall effect. We would finish our action potential faster. So the first 2 would affect our pacemaker potential. The last one affects our repolarization, all of which have the effect of increasing the number of heartbeats in a period of time, increasing the heart rate. Okay. We're showing that right here. So we have our action potential. We count from peak to peak within a certain amount of time. Okay. Let's talk about how that excitation is spread. So this happens in our pacemaker cells, but it has to go to all of our heart. And this is spread in a few different ways. But the effect is that the atria will contract first, and then the ventricles will contract. And so we're going to quickly spread that electrical signal, that depolarization through the cells of our atria. There will be a pause, and then we will spread it through the ventricles. And they have to be coordinated. You don't want different parts of your heart to be contracting at different times, right? You want it to be a coordinated effect. And this is through our intercalated discs and our conduction pathways. So an intercalated disc is a junction between 2 cells in the heart, and it has gap junctions that allow for the electricity to be spread, those ions to be spread. And it also has desmosomes, which are very tightly linked, and they resist mechanical stress. So here's what that looks like. We're going to spread cell to cell, starting in our SA node, right? Our pacemaker cells are going to fire an action potential. We then need to pass that depolarizing signal. So those ions, those sodium, calcium, mainly calcium ions, are going to be spread from cell to cell, cell to cell, through the gap junctions present in our intercalated disc. They are also spread in other ways. So we have signaling pathways that will spread the impulse as well. So an action potential is initiated in our SA node. That signal is going to spread through the atrial muscle via pathways called interatrial pathways. It will then be spread to the AV node, which is an area that's super, super important via something called the internodal pathway. I'll show you a picture with all of this soon. And a pause happens. So we're going to spread it to the atria, reach the AV node, pause, and then start to spread it to the cells of our ventricles, first by the atrioventricular bundle, sometimes termed the bundle of His, and it will then be split into our left and right bundle branches, and finally into Purkinje fibers. Let's see what this looks like. But first, the delay in the AV node. I want you to take a minute and think about this. Why is that delay important? We spread to the atria, we pause, we spread to the ventricles. This delay allows us to have a rhythmic contraction of our heart. It allows our atria to finish their contraction before we contract the cells of our ventricles. If you imagine the blood flow that we learned about last week, if we started to contract the atria and the ventricles at the same time, the blood would really get all messed up, right? We want to have that rhythmic circulation. So we want to contract our atria and then contract our ventricles. Okay, this is the conduction system I just talked about. It's going to start in our SA node. It will then be spread to the AV node via interatrial pathway, one atria to another, as well as the internodal pathway within the atria itself. Okay, then it will reach the AV node, it will pause, and it will then be spread via our AV bundle to our left and right bundle branches, split, and eventually split into Purkinje fibers. When it reaches the cells, this is where you're going to start to see that conduction through the intercalated discs, cell by cell, so that we have these areas of quick delivery and then our cell-to-cell delivery of that calcium signal. Okay, this is the same thing, but shown in a nice little pathway. So it's going to start in our SA node. It will then be conducted to the other parts of our atria. It will then reach the AV node and it will pause, and then it will rapidly be spread through the ventricles. Okay. Any questions at this point before we talk about another action potential? Let's talk about another action potential. I told you we're learning about 3 action potentials today. We've already talked about neurons. We know the neuron action potential. We've briefly talked about our pacemaker action potential that occurs right in our SA node. Now let's talk about what happens in the contractile cells of our heart. Okay, so we start the action potential in our SA node. We are then going to spread it to the contractile cells of the heart cells that do the work. And this is where the numbering system comes from. So phase 0 is going to be our fast action potential. Similar to a neuron, it's going to involve sodium. Then we're going to enter a phase 1, which is a repolarization phase. This might seem familiar. Oh, the sodium channel's closed, potassium channel's open. This totally makes sense. And then something weird happens. We have a phase 2 here. So we don't just go action potential, depolarization to repolarization. We undergo a weird plateau because now in the plateau phase, we open L-type calcium channels. We open a special type of L-type calcium channels, and all of a sudden there's a battle. The potassium channels are saying, let's bring this back down. Let's bring the membrane potential back down. I prefer the cell to be very negative because of my equilibrium potential, so I'm going to drive it in that direction. But the calcium channels are saying, no. I prefer the cell to be more positive. I would like it to breathe in that state. And what happens is the membrane potential plateaus. It plateaus for a period of time until the calcium channels inactivate and they close, and then the potassium wins, and we eventually get repolarization. That occurs in our phase 3, and we are at rest with phase 4. And this takes a long time. This is about a 300 millisecond action potential. So let's see that in action. So phase 4 is rest. Phase 0, driven by sodium, a fast action potential. Then we start in phase 1, get the brief repolarization brought by potassium. In phase 2, we've reached a plateau, a battle between calcium and potassium. Calcium inactivates, and in phase 3, we start the repolarization driven by potassium, and then eventually we get to rest. And again, this is a long process, okay? If we compare them, this is a comparison between the pacemaker cells, what we talked about in the SA node, and our contractile cell that I showed here. I'm not going to make you know. I'm not going to ask you to know. Make you know. I'm not going to ask you to learn. There we go. The action potentials in a bunch of different cells, but just know they exist, right? We're talking about 2 different flavors of action potentials in the heart. There are a whole bunch of them that have their own specific ions, their own specific slopes, all of that. We're really going to focus on the pacemaker one and this very stereotypical cardiomyocyte, where again, we've got a sodium current driving phase 0, and then a potassium current in phase 1, a plateau phase where there's a battle between calcium and potassium, and then calcium shuts off, potassium wins the day. We get a repolarization phase, bringing us back to rest. Okay, I would like for you to take 1 minute now. I would like for you to take 1 minute and draw on a piece of paper that you have hopefully sitting in front of you. Draw a pacemaker action potential, and then draw a cardiomyocyte action potential, and compare them, right? What are the differences? Duration, channels, right? Take a minute or two and draw on a piece of paper. And while you do that, I'm going to pull this up. See if you can remember. You can look back at your notes if you have to, but take a minute and do that. Now, Khan Academy has a great link, and this is in the PowerPoint, which is on the PDF page is on Canvas that has this link. Does a great job comparing these if this is something that is a very difficult concept for you, okay? But take a minute and draw. Compare the action potentials in pacemaker cells and cardiomyocytes. Draw a picture, draw a picture. Before I keep going, are there any questions people have? Going once, going twice.
Will you put this lecture on Canvas?
This should be on Canvas, yes.
Okay.
Yes. I'm also recording it, and I'll post the recording. Okay. I'm going to keep going. I know this is people's favorite thing. Okay. Let's talk about what happens next. So we focused on our pacemaker cells. They fire an action potential. They spread that electrical signal, that calcium signal, to the atria first, and then the ventricles via these conduction pathways, and then eventually through those intercalated discs. Now, how do we link that depolarizing signal with contraction? And that again is our excitation contraction coupling. And luckily, a lot of it is similar to what we talked about for skeletal muscle, and hopefully you learned in A&P. We're thinking T-tubules spreading that signal. We're thinking the sarcoplasmic reticulum is going to be involved with all that calcium. We have the same sort of myosin and actin filaments that have the troponin as the block. All of that is similar. If you learned about smooth muscle contraction, there are similarities there as well, because we're going to involve more calcium from the extracellular side, and we're going to spread it via gap junctions. But let's go through the steps here. And what I want you to think about is I want you to follow the calcium. I want you to follow the calcium. Our calcium is first involved in the L-type calcium channels here. Our calcium is first involved with L-type calcium channels. It will enter the cell. And that is, again, what gives us that early plateau in phase 2. A little calcium also comes in via these proteins called sodium calcium exchangers, but this is not a huge emphasis. Okay, so our calcium is going to come in via our L-type calcium channels. It will be detected by receptors on our sarcoplasmic reticulum called ryanodine receptors. Now, if you filled out the chart like you were supposed to for the pre-lab that we didn't, for the lab we didn't do, that chart about different chemicals, one of them you may have learned actually acts by binding as an agonist for our ryanodine receptors, and that is caffeine. So if you had your morning coffee, caffeine works in part, it does a few things. It binds to adenosine receptors, but it also binds to ryanodine receptors. And this is what effect calcium has on your heart rate. It speeds up your heart a little bit, right? Makes you a little loopy through the ryanodine receptors, okay? Calcium is, once it's released from the ryanodine receptors, now we have a wave of calcium. So calcium will first come in a little bit via these L-type calcium channels, and then a wave of calcium will enter the muscle cell from the sarcoplasmic reticulum. Those will bind to troponin, releasing that block, and will start the contraction process. They will then be put back into the sarcoplasmic reticulum by a protein called the SIRCA, or the SIRCA. So these are the key proteins involved. We have our L-type calcium channels. Calcium will come in. It'll be detected by our ryanodine receptors on the sarcoplasmic reticulum. They will release a wave of calcium. Calcium will eventually be put back by our sarcoplasmic reticulum calcium ATPase back into the sarcoplasmic reticulum. This will take energy. And the gradients will be maintained by our sodium calcium exchanger, which will then be put back where it's supposed to by the sodium potassium ATPase. So we maintain our gradients as well. Okay, we're going to follow the calcium. Let's think about it again. This is the same information, but shown a different way. We have our electrical current from a neighboring cell spread via a gap junction, right? It comes from the SA node, it goes via these pathways, and then eventually spreads into the muscle via these gap junctions, So our depolarization comes in. It is going to then move down the T tubule, right? All of this depolarizing current. Eventually, it will get near the sarcoplasmic reticulum, and it will be part of what helps release more calcium, right? So our calcium channels are going to open, our L-type calcium channels. We're going to detect that calcium at our sarcoplasmic reticulum with our ryanodine receptors. They will release calcium, and that flow of calcium will bind to troponin, release the block, and we'll get contraction via the sliding filaments here. And then calcium, we will spend energy to put calcium back into our sarcoplasmic reticulum and back into our extracellular fluid. So we'll use ATP to do both of those things. Okay. This is where we're going to end today. I want you to think about this question. What would happen if you added more depolarizing current? What would happen to our muscle contraction? What would happen if you added an agonist for L-type calcium channels? What would happen if you added an agonist for our ryanodine receptors, or if you added a SIRCA antagonist? So you guys have 10 minutes left. What I would like for you to do is I would like for you to draw me, you're not going to turn this in, but I would like for you to draw me an action potential in a cardiac myocyte normally, and then what would happen to that action potential if you added more depolarizing current, if you added more, an agonist for the L-type calcium channels, an agonist for the ryanodine receptors, a SIRCA antagonist, if we're affecting that process. And if the drug is affecting the cell, draw a picture of the cell. Draw a picture similar to this picture, right? Or this picture. And what effect is occurring via these drugs, okay? We're going to think about that. That's where we're going to end today's class. I'm going to briefly talk about the homework while you think about this question. And next week in person, we will pick up with that question. Give me 1 minute. Let me switch over here. So the homework assignment, if we are here, right? Module 2, pacemaker cell homework. We're going to do part 1 and 2 of a case study. So here it is. It's asking you very similar questions, if it will download. Here it is. Okay. You're going to use words from this word bank to think about the contraction of a heart, okay, in part 1. Then You've got more things for your word bank. Please put all that in. You're going to draw an action potential, draw ion movement, all these things we've been thinking about. And here are the graphs for that. And then in part 2, you're going to start to think about these symptoms, and I'm asking that you go to this website and try and determine what's happening in parts 1 and 2. We will next week work on parts 3 and 4. So to end class today, I want you to think about these questions. Next week, we're going to pick up right where we left off. And we're going to think about what happens to the heart if we add different drugs. Are there any questions at this point? Okay, I know I threw a lot of material at you. What I want you to do is I want you to spend the next 10 minutes going back through your notes, right? Writing what you learned, maybe drawing some pictures, all the things we would do in a normal class if we were face-to-face and I had you in front of me to draw me things. Take 10 minutes, draw pictures, think about the steps, think about the effect chemicals will have on these steps, and we will pick up with that in person next Wednesday. With that, you guys are good to go unless you have any questions.